Great. And I think let's see, yes, the next speaker is Cesar Serrano, who's a medical oncologist from Barcelona, who's with us doing research on just one of the things, again, that the GIST Cancer Research Fund has allowed us to do over the years is bring talented individuals who are committed to making progress in GIST and sarcoma from other parts of the world and bring them to Boston, to Harvard for a period of time for concentrated study, at which point they then go back to their home centers of excellence and start up an enduring research program which leverages everything that GCRF is supporting and everything that we're doing. And Cesar, who's early on in his fellowship here, is a nice example of that.
Thank you, Jonathan, for this kind introduction, and thank you, everybody here, for being here today and for organizing this nice thing. So, the goal of my talk today is to tell you about our work in the lab, where is our understanding of the biology of the GIST right now, and what are we working in the lab trying to improve all this understanding over time, always with the goal of translate all this knowledge to the clinical management. So, as you may know, there is a continuum in GIST tumors and other sarcomas and other cancer types, from early lesions to symptomatic disease that can be either localized or metastatic. So when the disease is localized or metastatic, we start to treat it. And we use surgery, we use drugs, and eventually in the same patient, they develop resistance to these drugs. So this is why probably the main focus in the most of the labs that do cancer research is in resistant disease. Concerning GIST, we have to bear always in mind that the most of the GIST, or the proliferation of the GIST, are driven by KIT. Okay? So there is a great treatment for KIT, for this GIST that bear on KIT mutation, that is imatinib. So we have different types of treatments in different timings, like surgery, like imatinib in a metastatic disease. But eventually, same patients develop drug resistance, imatinib resistance. So we have still a few options, like increasing the dose of imatinib, use sunitinib, and other options that we can use as in clinical trials, for instance. But there is quite some patients that develop this drug resistance. This is just a few numbers to show you. This graph comes from an updated results from the original clinical trial that led to the approval of imatinib. So you can see here that around 50% of the patients do quite well during the first 2 years, but eventually over the time, all the patients, not all the patients, but around 80% of the patients develop resistance to imatinib. So since this is the most concerning thing that we see in the clinic, is what we are trying to overcome in the lab with all the investigations. So what can we do so far in GIST? How can we treat it? If you pay first attention in this picture on the right, you have here the KIT receptor and the KIT pathway. Here is the membrane, the receptor, and here is the nuclei, the DNA, right? So KIT receptor signals through these two main pathways, that's PI3K and RAS pathway, to stimulate the proliferation of the cell. So how can we treat it? First of all, we can target KIT directly, and we can do it with imatinib, sunitinib, hopefully soon with regorafenib. We can do it as well with this new generation of KIT inhibitors that are under development. We can also target KIT with other inhibitors through more complex mechanisms like CDC37 inhibitors or HSP90 inhibitors. But you also can see that if these two pathways are very important, we can target both of them either together or separated. But we have to improve this, of course. So what can we do? That means, what are you guys doing in the lab? So in the lab, we start always from the beginning. And the beginning for us is always the clinical patients and the clinical care. So we usually, when we visit patients, we came up with some questions. And we don't have the answer at all. Even you, when you are in the clinic, and tell us any question, and we don't know what to ask to tell you. So some of these questions, we still can test them in the clinic. in the lab, right? So in the lab we have cellular models that resembles quite a lot to the biological behavior of the tumors in the patients. So here in the lab, what we do is just treat these cells with, for instance, with drugs, and see what changes do we have in proteins or in genes. So once we have a hypothesis of this question that we had in the clinic, we go back to the clinic. And we try to test it before with clinical data coming from pooled patients and also from tissue specimens coming from biopsies or from surgical samples. And if we still have some doubt, we come back to the lab and try to answer it much better than we did, and then go back to the clinic again with always the end point to try to change the patient care. So just be sure that through this continuous feedback between the clinic and the lab is the best way to improve the knowledge and the understanding of the gist. When I was in the medical in my degree, doing my degree in medicine, so I thought that it was quite easy. I imagine a cell something like this. We had here the KIT receptor, and here was the pathway telling to the nuclei, to the nucleus, to the DNA that okay, you have to grow, and you have to grow, you have to grow, and you can easily just beat it. Just put here something, block this pathway, put an elephant or whatever here, and just block the road and cure the cancer. But the problem is that the more you study the biology of the cancer, the more you realize that it's more complex. So right now, I have other thoughts about these cells, like a crossroad, all right? So we have down here the KIT receptor sending proliferation signals through all these lanes, all these roads. But there are also other many roads that we don't know exactly the function, or even we don't know what are they doing, all right? So it's much more complex than we thought previously. But it's still interesting because we have some tips for that. For instance, we know that imatinib is a great drug. So it is one of the best drugs ever in cancer. But we need something better. Something better means super imatinib, or means combining imatinib with something else to overcome resistance to imatinib, okay? But the other thing that concerns a lot of to us in the lab is how can we study all this mess? So the traditional way to do it was interrogating specifically each pathway, each lane of the road. But it got us very good results. But right now, all the science is moving through high-throughput techniques. That means trying to interrogate all these pathways at the same time, all these lanes of the roads, to see what happens and create a comprehensive and integrative knowledge of what is going on with these cells. So we can do it by two approaches. One is through genomic, and the other are functional screenings. So genomics is a kind of descriptive, but we can get knowledge of the G cells as well, other tumor, of course, other tumors, but very in detail a lot of genes at the same time, not studying specifically one gene and then other gene and then other gene that will take us for years. So it gives us a lot of information from the beginning. But the other very interesting approach are functional screenings. And this can be with loss of function and gain of function. So loss of function means just we block one gene and see what happens. And gain of function means just we try to stimulate one gene and see what happens in the cell. That means the cell doesn't care, the cell proliferate more, or the cell die. Okay? So we are getting ready right now the gain of function approaches, but we have done a lot of work in the lab with these loss of function approaches. So loss of function approaches basically means that we interrogate many genes at the same time in the cell. It's just not a few. We are talking about thousands of genes, trying to look for synthetic lethality approach. So what does synthetic lethality approach mean? I will try to do my best because it's not easy sometimes. So we come back to the gene-resistant cell. We have the KIT, and we have all the proliferation stimulate telling to the cell that the cell has to grow. So if we block the road here with imatinib, you can see that there are many proliferation signals everywhere that are still telling to the cell that the cell has to grow, all right? So just imagine that we are working in the lab and we find other cool pathway that say, wow, this might be important for a yeast cell. And we treat this yeast and this pathway, and okay, we block this pathway, but we still see proliferation in the cell. So we maybe can think that it's not important at all, but we cannot be right. So this is the concept of synthetic lethality. We have two important pathways in the cell. If we block both separately, we don't have any effect, but if we combine both, we just inhibit the proliferation of the cells. And this is what synthetic lethality means, and what I try to mean with this graph. So we have these two pathways, the KIT pathway that is already known, and we have the other unknown pathway that even we don't know. So when we combine both, we kill the cell. And the best way to identify this novel target are these loss of function screenings that we have been doing and that we are still doing in the lab. So this is a fantastic work doing for my colleague Adrian as well. So what we have is just the yeast, right? So we have untreated cultures of yeast and treated cultures of yeast with imatinib, and we infect them with viruses. So these viruses, what they do is just join to the genes and avoid the expression of the genes, just knock down, blocking them. So finally what we do is just harvest all this DNA, amplify, and try to see what happened to the cells and to these shRNAs. If the shRNAs just were like this, or what they didn't change, or if they impact in the proliferation of the cell somehow. That is the important thing for us. So it's not just getting a ranking of these 11,000 genes, right? It's just studying all these genes, because as you can see, these pathways are very complex. So there are many units in each pathway that we have to try to put together and to see the significance of each gene. But okay, you can see here one of these data set, the top hits, and this is one of the achievements of this pool screening is CDC37 that ranked the best one in essentiality of the cell. So what we validated this result, we got that with blocking the CDC37, the KIT expression was gone, also downstream pathways. And when we went to the cell lines, you can compare these 4 panels with these 2 panels. These are untreated cells, and these are cells in which we block CDC37 with shRNA inhibitors, and we see the lack of proliferation of these cells when we inhibited CDC37. So through these experiments, we saw that CDC37 is very important for yeast cells. If we block it, the cell die. And CDC37 is also very interesting for future clinic approaches because it's a cofactor of HSP90. So it's a kind of HSP90 but very specific for kinases like KIT. So we probably can say that CDC37 inhibitors will have less toxic effects than HSP90 inhibitors. But there is no CDC37 inhibitor yet in the clinic. But very, very interesting approach that we have got through the shRNA screening. Other knowledge that we are getting through these shRNA screenings are understanding the KIT pathway. As I said before, we have here the KIT pathway, and I always compare with a kind of a Greek temple. Okay, we have the Greek temple of KIT, or the Greek temple of the yeast proliferation. So to avoid this temple to collapse, we need KIT up here. Actually, the cell needs KIT up here, but also needs these 2 important columns here. One is the PI3K pathway, and the other is the RAS pathway. So we can collapse this temple if we just destroy KIT, but also it's very important or meaningful for the cell if we destroy PI3K or we destroy RAS. And this is what we are working with right now in the lab. So going with the RAS pathway, okay, the MAPK pathway, we look through the data sets and we see that there are some meaningful hits along the data sets, and all these hits are important in the RAS pathway, in the MAPK pathway, that suggests us that this pathway can be meaningful for yeast. But we have to prove it. That's not enough, just having these data sets. So what we did is blocking the MAPK pathway. And blocking the MAPK pathway with a MEK inhibitor, we see that the proliferation was much reduced compared with imatinib or compared with sunitinib if you compare them with the control cell that untreated cells. If we go to the cell culture, these are the control cells untreated, and these are the cells that was treated with the MEK inhibitor. So we got the evidence through the data sets coming from these loss of function screenings, but we prove it with some drugs that we have in the lab. And in the same way, we went as well to the PI3K pathway, and you can see that there are important hits through all the pathway of the PI3K pathway in GIST. So we did PI3K inhibitors, or inhibition of this pathway along with other kinds of inhibitors, and we see the same results that we got with the MEK inhibitor. But we came up with an interesting idea. The most of the experiments that we perform in the lab last like 3 or 6 days. But if you use one of these inhibitors in clinic, it lasts months or years. So we wanted to know if we treat one cell line for a long period of time, what happened with a PI3K inhibitor. So we can have two options. One option is the cell won't care, and there will be other pathways, other roles that will circumvent it, this pathway, this PI3K pathway, or well, we have this PI3K pathway that is very important for the cell, and the cell had to fix it somehow just to get activated this cell. So we treat this G7882 cell line with a PI3K inhibitor, one of these both two columns, for 6 months, and after that we got a different line, something completely different that you can see here from this previous and after these 6 months of treatment. So what the cell did, I mean, just don't care too much about this. This is AKT. This is that means activation of the PI3K pathway in the original cell line. After these 6 months, what the cell line did was fix it. Just lost this P10 and reactivate the AKT pathway. See here the greater phosphorylation. So through this experiment, we can say that PI3K pathway is very important for GIST, as is the MEK pathway. So we just have the loss of function screens. We see that there are many hits. They may be important. We treat them with some inhibitors. We see that they have effect. And then we treat the cells for a long period of time to see what the cells do. And what the cells do is fix these pathways. We have this model in PI3K pathway inhibitors, but we also have this model with RAS inhibitors. So these are basically the greatest hits that we are getting with our shRNA screens, and all the things that we are working with, because if we treat the patients right now with PI3K inhibitors, they are very likely to end with resistance at some point in the evolution. And our final goal is trying to find a drug or a drug combination that will eventually avoid this resistance. So just to summarize, the main points of the talk is that these new high-throughput technologies allow testing many potential drug targets in GIST simultaneously, which is very important for our knowledge and for the patient management. And always the final objective, our endpoint or the experiments that we are doing, is trying to maximize imatinib efficacy to eradicate these resistance of clones. And just thank you to you very much for paying attention, and for all the people from DNFR and from the guys from my lab. Thank you very much.